Chloroplast structure. Chloroplasts are organelles found only in plant cells that are essential to photosynthesis, as they contain chlorophyll, which captures, converts, and stores sunlight while releasing oxygen. The chloroplast has an inner and outer membrane with an empty, intermediate space in between. The inside of a chloroplast contains thylakoid discs, which are stacked, called grana, as well as stroma, which is the dense fluid in the chloroplast. These discs contain the chlorophyll used in the process of photosynthesis. The space filled by the chlorophyll is called the thylakoid space. The lamella is a plate of tissue that connects the thylakoid discs and helps to maintain the structure of these bonds. The thylakoid membrane is an internal system of interconnected membranes that carry out the light reactions of photosynthesis. They are arranged into stacked and unstacked thylakoids.
This cycle starts by a photon of light striking the antenna pigment molecules that are found in photosystem II. The energy that comes from the photon is transferred to the reaction center of this photosystem. This excites the electrons, causing them to move up an energy level and are replaced by other electrons from water. This energy is also used in photolysis, which splits the water molecules into protons, which are stored by the thylakoid lumen. Oxygen is released into the environment from the leaf and electrons. These are all transferred to the electron carrier 1, which transfers them to their correct locations. The H is transferred to the stroma, and the electrons are transferred to the secondary electron acceptor. The electron carrier 2 then transfers them to photosystem 1 when needed. The same process happens here again. Other electron carriers transfer them from the reaction center of photosystem 1 to molecules of NADP, which allows this NADP that was released from the Calvin cycle to combine with those H ions that are found in the stroma. This forms NADPH, which is stored in the stroma to be used later in the Calvin cycle.
The Calvin cycle. This cycle produces molecules of G3P in the stroma of chloroplasts. To produce one molecule of glucose, this cycle needs to be repeated 6 times. The Calvin cycle reactions are also called light-independent reactions because they are not directly driven by light. The Calvin cycle has 3 stages. Step 1. Carbon fixation. A CO2 molecule combines with a 5-carbon acceptor molecule, ribulose 1, and 5-biophosphate to create RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase/oxygenase, or Rubisco, which is how the carbon dioxide molecule is fixed. This step makes a 6-carbon compound, which is initially unstable. It splits into 2 molecules of a 3-carbon compound called 3-phosphoglyceric acid, or 3-PGA. It is now stable. Step 2. Reduction. The new 3-PGA molecules are activated by ATP, then reduced by NADPH, a reducing agent in the Calvin cycle to reduce atmospheric carbon dioxide, producing monosaccharides for use by the plant for structure and energy to form 2 molecules of G3P. If allowed, the G3P molecules leave the cycle instead of regenerating into RUBP and can be converted into glucose in the cytoplasm of the plant cells. You need 2 molecules of G3P to create 1 molecule of glucose. Step 3. Regeneration. Energy is supplied by ATP formed during chemiosmosis. This energy is used in a series of chemical reactions that break down and reform the bonds needed of the G3P molecule so it can regenerate into RUBP. To summarize, in stage 1, the enzyme Rubisco incorporates carbon dioxide into an organic molecule. In stage 2, the organic molecule is reduced. In stage 3, RUBP, the molecule that starts the cycle, is regenerated so that the cycle can continue.
Chemiosmosis. To begin, protons, or hydrogen ions that come from photolysis, are collected in the thylakoid lumen. These travel down the concentration gradient from the thylakoid lumen to the chloroplast stroma through diffusion. They pass through the enzyme ATP synthase that is located in the thylakoid membrane. ATP from the Calvin cycle is converted to ATP by chemiosmosis. This enzyme rotates. Because the hydrogen needs to move, the ATP synthase uses the transportation of this hydrogen to rotate the ATP synthase, which creates the ATP through the combination of inorganic phosphorus and ADP. This happens on the stroma side of the membrane. The hydrogen then travels to the stroma. The molecules of ATP are used in the Calvin cycle.
Cellular respiration. In cellular respiration, there are 4 steps through this process. There is also something very important called the electron chain. One thing that happens numerous times to keep in mind is that there are 2 molecules called NAD and FAD. These molecules often carry electrons, so when they pick up these electrons, they gain 2 protons and they become NADH and FADH2. So when these are mentioned, make sure you understand the process behind that.
First step, glycolysis. In the first step of cellular respiration, there are multiple chemical reactions. Every organism, not just plants, perform glycolysis. This process happens in the cytoplasm. One reaction, which you all know, would be the reaction that breaks down glucose and oxygen into carbon dioxide and water. Let's go through it in terms of the process of glycolysis. Starting with glucose, 2 molecules of ATP are used, which produces 2 molecules of pyruvate, 4 molecules of ATP, and a molecule of NADH. Pyruvate is a Greek name for sugar splitting, which makes sense because in this reaction, our 6-carbon sugar, glucose, went through a reaction that gave us 2 3-carbon sugars, pyruvates.
The second step, pyruvate oxidation. This step is one of the smaller ones, creating a bridge between our final step, glycolysis, and our third step, the Krebs or citric acid cycle. Most cycles in cellular respiration happen inside the mitochondria, but our first cycle, glycolysis, starts in the cytoplasm. Pyruvate oxidation is the reaction that brings the whole process into the mitochondrial matrix. We start with the pyruvate molecule from our previous step. This 3-carbon sugar drops 1 carbon molecule as CO2 and changes to a 2-carbon molecule called acetyl. The energy that we've been carrying since the first step with the glucose is now in the acetyl. The CO2 that was dropped now has electrons available for use. They can't be taken out of nothing, so a molecule called NAD picks up that electron and becomes NADH. Now the acetyl needs to get into the membrane. To do this, it binds with something called CoA to complete this transport. This creates acetyl-CoA, which is accepted by the membranes into the mitochondrion. After this is completed, the CoA is dropped as it is not needed anymore, and we are left with acetyl, which brings us to the next cycle.
Third step, citric acid cycle. In this next cycle, we're going to start with a 4-carbon molecule called oxaloacetate, which is OAA for short. Taking our reactant from the previous step, the 2-carbon molecule acetyl, we add it together with OAA to create our first product, citrate. In the next reaction, the citrate loses 2 CO2 molecules. 2 NADH captures the electron from this dropped carbon. Then there is a series of many reactions. Through these reactions, first, ATP is formed, then we see more electrons that are removed by something called FAD, which gaining electrons turns into FADH2. Then lastly, we see electrons being carried again, but this time from a molecule called NADA, when gaining electrons turn into NADH. Now we end up back at OAA, completing the cycle. The purpose of this is getting energy and electrons, and we end up with 3 NADH, 1 FADH2, and 1 ATP to achieve that.
Fourth step, oxidative phosphorylation. This includes the process of NADH being oxidized into NAD plus hydrogen plus 2 electrons. So, our carrier has now done its job, and these electrons must be accepted. You can look at oxygen as the receptor because half an oxygen molecule combines with the 2 electrons and 2 protons to result in the creation of H2O, or water. In every step of this reaction, energy is being released. The electron chain is essential for this step because it's what allows our protons and electrons to end up in the end reaction.
After having absorbed high-energy electrons all throughout cellular respiration, carriers such as NADH and FADH2 must be able to release those electrons. Oxidation is the process where an oxygen molecule gains electrons and combines with hydrogen over a series of processes to produce water while continually releasing controlled energy. The electron's energy is transferred to proteins within the inner membrane of the mitochondrion, which pump out hydrogen ions into the intramembrane space. This concentration gradient of protons is similar to that during photosynthesis, and an ATP synthase lets the protons pass back into the membrane while creating ATP from ADP and inorganic phosphorus.